Thanks. Okay, I'm going to trust everyone on the call can hear me, but we won't know if you can't. So I'll plow forward. If I look a bit uncomfortable, it's because I just cycled in and then squeezed into a suit, so I am actually a bit uncomfortable. I'm sorry about that. I have been now in the Department of Urologic Sciences for 11 years. Actually, I came in 2011, like a 3-4 year postdoc, and then I've been faculty since 2015. And yeah, but Peter's right, like during that time had this arc from basic genetics, which is what my PhD is, all the way through to now sort of computational biology, which is what most people in my group do, although I cannot. So sort of a supervisory role there. So what I'm going to talk about today is a range of different scenarios where circulating tumor DNA is poised to impact the clinic. And we'll start with detection, minimal residual disease, just because that's kind of what a lot of people are aware of. But I'll be talking a lot about our own work in the really late setting where we're trying to characterize the genomic alterations in ctDNA and then use that to try to select therapy and sort of optimize treatment. Here are my disclosures. So I think as many are aware, the term liquid biopsy is really this like all-encompassing term that kind of refers to just
Is it going down anymore?
It's off. Is that better? Yeah, that's better. It's sort of this all-encompassing term that kind of refers to any type of tumor material shed into body fluids. So that could be blood, urine, spinal fluid, all sorts. What I'm going to focus on today is circulating tumor DNA, which is DNA from the tumor that's shed into the bloodstream or other body fluids. So ctDNA is really short fragments of tumor-derived DNA in the blood. It's gone through apoptosis, so it's been already chopped up by the machinery of the cancer cells. And when it's in the bloodstream or the urine, it's mixing with cell-free DNA from normal cells. So in the blood, that means dying white blood cells. In the urine, that will mean cells of the urothelium and so forth. It's got a short half-life because it's cleared by kidneys and your liver and enzymes in the blood. And this fourth point is really important because it's a bit of a disconnect. Initially a challenging thing to grasp because you think, well, it's coming from dying cells. Well, the best analogy is actually in pregnant women. And we know that by just 10 weeks gestation, about 20% of the maternal blood DNA is derived from the fetus or really the placenta. And again, we think of that as a proliferative, rapidly invading entity at that point. Right? So the amount of ctDNA in the blood is closely associated with proliferative tumor burden. So just a reminder that the ctDNA is tumor-derived cell-free DNA. So you'll hear about cell-free DNA and ctDNA, and the difference is really cell-free DNA is everything, and ctDNA is the part that's tumor-derived. And we can tell it's tumor-derived because it carries things like mutations, base changes, copy number changes, structural rearrangements that we don't see in all the matched white blood cells that we're profiling, the normal from that patient. We've shown recently that if you look within ctDNA, it's much more diverse than if you profile a single metastatic tumor. There's really loads of ways to show this. This was a figure we published this summer. You don't need to understand what this shows to see that the tumor tissue plot is much cleaner than the ctDNA plot, the cell-free DNA plot. And it's because the cell-free DNA is containing lots of different populations, and they're kind of all contributing different levels of alterations. And ultimately what it means is that the contribution of any individual metastatic foci, or really any tumor foci in general, to total ctDNA is actually quite low. And ctDNA together is derived from lots of different metastases in a body. So the other thing that's sort of important to bear in mind before I go forward with some of the utility is that this is kind of conventionally what people think is present in cell-free DNA in an advanced cancer patient, where you've got your normal populations from dying white blood cells and you've got your cancer populations from multiple sites. But in fact, there are other things in there too. You've got normal cell-free DNA from non-hematopoietic tissues, potentially linked to comorbidities of the patient, from treatment side effects, from the tumor microenvironment. You've also got clonally expanded white blood cell populations, so essentially hematopoietic expansions of indeterminate potential, and potentially you also have other subclinical cancers in there. So whenever you're designing approaches to interrogate ctDNA, you have to be aware of all these other things in there and make sure that your experimental approach kind of controls for them. So I'm going to run through this through a few different areas where ctDNA tests are poised to help in the clinic. They're listed here, and I'll run through them one by one. Well, to be honest, actually, I'm not going to go through improving cancer screening in at-risk populations. That's kind of for another day, perhaps. I think the key distinction as we go forward is that there are really two main classes of ctDNA tests that are under investigative development right now. The first is about just detecting ctDNA. So if you can find ctDNA, it normally means you have cancer somewhere in the body. And so that's things like improving screening, detecting residual disease, potentially monitoring for response or resistance. But then in the later setting, the individual already has cancer. So now you want to actually characterize the alterations that are present and sort of understand the biology, the genomic makeup of the tumor. So that's when you need a different class of assays to actually tell you what's happening within the ctDNA. And I would say it's a little bit cut off here, but the lack of recognition between these two types of tests is contributing to some unrealistic expectations of sensitivity in a late stage setting, because you can be really, really sensitive to detect ctDNA, but you may not be able to actually characterize it. And so many in the MRD field, the residual disease field, are looking at the late stage setting and going, you guys, your assays suck. But the reality is, as I'll explain, it's because the sensitivities are not possible to get at the moment in a cost-effective manner. And it's because of this. So clinically pragmatic assay design, you've always got to have cost in mind. Now, in an ideal world, what you do is sequence the entire genome thousands and thousands of times. You'd look at every single copy of DNA that was in your tube. But that would cost millions of dollars. So we can't do that. We have to compromise, and we say, we're going to look at only a subset of the genome, and we're going to look quite deep at that subset, right? And obviously, the fewer parts of the genome you look at, the deeper you can go. So there's really two types of assays. One is tumor-informed. So that's when you have, let's say, a cystectomy specimen from an individual. You've profiled that tumor, that primary tumor specimen. You know a handful of mutations that are present in that patient, and then you look for them really, really deeply in the blood. So you're really just looking at a handful of pieces for DNA. So you can spend all your money going very, very deep. So that is pretty much where the leading edge for bladder cancer development is at the moment for tumor-informed assays. For the other class is tumor-agnostic. You don't know what's going on in your person's cancer. So typical of, let's say, late-stage prostate cancer. And so here, you can't just look at a few points, because what's the chance you're actually going to pick up a mutation in those few regions? So now you have to look across a lot of DNA. But you can't do that as deeply unless you have an infinite budget. So that's where you have lower sensitivity later on if you're trying to understand what's there de novo. I hope that makes sense. So the first use I want to talk about, and this is particularly relevant for bladder cancer, is detecting residual disease. So here's a retrospective study. It was locally advanced bladder cancer given neoadjuvant chemotherapy, cystectomy, and then surveillance. Now, in the study, ctDNA detection after cystectomy, you can see here recurrence-free survival and overall survival. So ctDNA detection in the red really was indicating imminent relapse and obviously worse prognosis. And the median lead time between detecting ctDNA and clinical relapse was more than 3 months. So quite a big advance forward. So, obviously this gives rise to the hypothesis, is there an opportunity for early intervention before clinical relapse in patients after cystectomy that shows ctDNA positivity? And there is actually a trial going on by Lars Dierscott's group in Denmark to test this hypothesis. So similarly, in the neoadjuvant setting, there was the Abacus trial published in European Urology this year, or at least the final analysis was published in muscle-invasive bladder cancer. And here, the presence of ctDNA, again indicated in red, associated with shorter recurrence-free survival, both pre-treatment, after treatment, and after surgery. So again, indicating that, you know, the continued persistence of ctDNA is a poor prognostic indicator. And potentially those that have very low ctDNA, you know, there may be opportunities to test whether those individuals are, you know, may not need actually cystectomy in that case. And then the final bladder trial I'll mention was in Vigor 10, and this was a randomized phase 3 of atezolizumab versus observation after surgery, so in the adjuvant setting for muscle-invasive bladder cancer. This trial was actually negative, didn't meet its primary endpoint, but there was it was noted that there was considerably longer disease-free survival and overall survival for ctDNA positive patients that were treated with atezo. So you can see that at, the laser's not working very well, but the bottom set of curves on both sides, those are your ctDNA positive patients. And you can see that they seem to do better with adjuvant atezo compared to observation. So, you know, does that suggest that maybe ctDNA could identify patients who are at increased risk of relapse and may benefit from adjuvant treatment? Yep.
Do they show reversal of the CT positivity? Does it become CT negative after the treatment?
Yeah, so that, again, yeah, that you're right. After a couple of cycles, there are some patients that show clearance, and those have particularly prolonged disease, essentially control, yeah. So the second thing I want to talk about, the second area, is to estimate cancer aggression, i.e. prognosis. So the first point I'll highlight is that ctDNA purity is variable in metastatic cancers and probably in local disease as well. So previously, I've just been talking about presence-absence, and now I'm showing you that actually when we look, this is 4 different cohorts, well, 3 profiled by my group and 1 by an external group, showing the fraction of ctDNA in the blood, so the proportion of your total cell-free DNA that's tumor-derived. And so these are 4 different metastatic prostate cancer cohorts, and you can see this big range from 0 to something like 90% in some patients. 90% of their blood DNA is tumor-derived. And then on the top right is a cohort of metastatic bladder cancer that was put together with Peter and Bernie Eigel at B.SC Cancer. And again, you can see this huge spectrum. Now, we have shown that generally metastatic burden is associated with this range. So those patients with 80-90%, they'll have visceral disease, you know, widespread other indicators of heavy tumor burden. But it's not actually a perfect predictor. And there are plenty of scenarios where you wouldn't necessarily expect high ctDNA, and maybe you're just seeing the explosion of disease happening. But what we've shown across, this is actually 2 randomized phase 2 trials led by Kim Chi in Vancouver, that high ctDNA in a metastatic CRPC patient prior to treatment is associated with really poor prognosis. So this deep red line is above 30% ctDNA. This is kind of your middle level, and this is very low. And we're looking at overall survival after first-line CRPC in an unselected population, and in the middle in a poor prognosis population. And so again, you can see like very robust splits between the Kaplan-Meier curve just based on the amount of ctDNA in the blood, right? This is a simple, relatively simple to measure variable. And on the right-hand side is a bladder cancer cohort, first-line metastatic UC. Most of these patients were subsequently treated with platinum, but again, you can see a big split between high and low ctDNA. So I think the take-home message is that high levels of ctDNA in a patient with metastatic cancer, probably in general, means short life expectancy. And there's actually ongoing work in my group in collaboration with Kim's to do a meta-analysis, pulling together as many patients as we can in the first-line setting to actually understand exactly where these hazard ratios lie for patient prognosis. And this particular data freeze here, this is 463 patients about to initiate first-line treatment, and the hazard ratio for greater than 30% there is 5.7, and actually that's independent of all clinical prognostic markers. So we're preparing this for publication at the moment because we think that this is a relatively easy to measure blood variable that's actually being reported by many clinical tests now, and so I think something people should be aware of. The third scenario I'll talk about is now you've begun to treat your patient with metastatic disease, and can ctDNA offer an opportunity can it provide a surrogate for response? So we noted a few years ago, actually leveraging some patients from the urology clinic here, that in newly diagnosed metastatic prostate cancer, initiating hormone therapy, these first few days after starting Tigerelix in this case, you can see quite rapid declines in the amount of ctDNA in the blood just in those first few days. So suggesting to us that effective treatment is very rapidly impacting the amount of ctDNA in the blood. And again, this gets back to the idea of it coming from proliferative lesions, because obviously we'd be expecting log kills right now in terms of the cancer, and why aren't we seeing spikes in ctDNA? So we've actually been testing this in the later setting, and this is actually a collaboration with a group in the Netherlands, and as part of a prospective observational trial, 81 patients initiating first-line enzalutamide or abiraterone for their CRPC. And we had a blood collection at baseline before they started treatment, and then just after 4 weeks. And what we saw was that after 4 weeks, there are some patients that had high ctDNA at baseline, went to undetectable by 4 weeks, and others retain this persistent ctDNA. And look at the split between those curves. Just 81 patients just after 4 weeks, and a hazard ratio approaching 5 there for the red curve. So again, here we think that I should mention, even compared to PSA at 4 weeks, this showed considerably stronger performance, sort of positive predictive value, negative predictive value around 90%, for identifying patients that don't benefit long-term from treatment. So we think that on-treatment ctDNA dynamics are a surrogate, will be a surrogate marker for progression-free survival. And I would also mention that later on, so this is after 12 weeks of treatment in patients with poor prognosis disease, we can now see that actually rises in ctDNA on treatment are again indicating progression. So I think the general picture we're getting is that we're kind of seeing that the molecular changes in the blood precede the clinical presentation associated with responses or resistance. So the penultimate category I'll talk about is prediction of treatment efficacy. And this is a kind of longer block because it contains a lot of work we've done in this setting. I think as many are aware, when we look at prostate cancer in general, particularly in the metastatic setting, you'll often hear this like genomic heterogeneity, every patient's different, and so on and so forth. But the reality is that you can actually group patients' tumors together on the basis of the type of alterations they harbor. They may not have the exact same changes, but they have changes within the same classes of pathways, same types of pathways. You can kind of form these rational subgroups. And so, for example, we know that there's a group of patients that have DNA repair defects, whether that's in BRCA2 or ATM, mismatch repair genes, so forth. And we actually have consensus clinical guidelines around the world now that even recommend searching for this subgroup that have DNA repair defects. So we and others have shown in several correlative studies that each of these kind of classes, as well as others, have different clinical relevance in the context of different treatments. So both prognostic implications and also some predictive implications, and I'll walk through a few of them. The first point I should make is that reassuringly, when we look at genomic alterations that are present in ctDNA, we find that by and large, they're the same that we would find in matched tissue from the same patient. And I think this can actually be a failure of many liquid biopsy studies, especially outside of ctDNA, in that the key thing you've got to do initially is to ground the stuff you're seeing in the blood in the tissue, right? Remember that tissue is the gold standard. And so any observations you make in the blood probably need to first be observed in the tissue, to be taken reliably. So in the ctDNA, we see the mutation profiles between matched ctDNA and tissue, very concordant. Copy number profiles also very concordant. We've seen this across prostate and bladder cancer as well. And to give you an example, actually from bladder cancer, the alterations in ctDNA do seem to reflect clinically relevant tumor characteristics. So this slide here is showing metastatic UC with FGFR3 or ERBB2, that's the HER2 gene, with those alterations identified in ctDNA. So in the plots on the bottom left, that we're now looking at the RNA-seq expression of the FGFR3 transcripts in tumor tissue. Now, the patients where we identified mutations in the ctDNA have considerably higher expression of FGFR3 than those that were wild type in the ctDNA. The same is true for alterations in HER2, and this corresponded to high protein expression in the tissue. So what this is telling us is that when we find, in this case, an FGFR3 mutation in the ctDNA, that seems to be reflective of an upregulated pathway in the tumor tissue. We would hope this to be the case, but it's important to sort of show this and reassure yourself that these really are reflecting biologically relevant events. Similarly, here's a really unusual bladder cancer case that appeared to have a biallelic deletion of BRCA2. So this is a gene that repairs double-stranded breaks. And this is quite rare in bladder cancer. We know that PARP inhibitors have not shown efficacy in unselected metastatic UC, although platinum obviously does have a role. But we don't widely consider this disease to have homologous recombination repair deficiency, and yet here was a case that appeared to have it. And when we looked across the whole genome, we could actually see the mutational signatures that are associated with HRR deficiency. So this was really a smoking gun, that this was one of those rare tumors that had genuine HRR deficiency. In prostate, we know that there are several different classes of DNA repair defects. So although I've just said we can group all these tumors together, the reality is each of these different types of alterations has different downstream effects. And I'll show you why this is relevant. So here I'm showing the four major classes of repair defects in prostate cancer: CDK12, BRCA2, ATM, and then mismatch repair deficiency. There's a constellation of genes that contribute to this. So just visually, we're looking at copy number profiles from 4 representative tumors. You can see they look different, right? These are kind of, they have just a different phenotype, and this manifests even grossly at this level. So CDK12, poor prognosis. BRCA2, poor prognosis, but good outcomes with PARP inhibitors. ATM seems to be neutral or good prognosis. MMR deficient, good outcomes with immunotherapy. These relationships have been shown by us and others around the world as well. And so what this means is that the specific type of DNA repair defect you have has differential implications for both patient monitoring, decision timelines, and ultimately even treatment selection in some cases. So we're talking here about not just prognostic indicators, but predictive indicators too. And of course, BRCA2 defects are indicated. Oh, you have a question about that?
This question for you. You get mutational heterogeneity across tumors, primary and the metastatic. I'm wondering, does this profile, is it weighted towards the primary, weighted towards the metastases, or how does it represent the overall mutational profile?
Mm-hmm. So the question is whether this type of profile I'm showing on the screen would be more representative of the primary or the metastases or one region of the tumor. So in the ctDNA, you're getting a kind of bulk And as I'll mention later, we can actually deconvolute that a little bit to see what the different populations are. DNA repair defects tend to be very truncal. They tend to be quite early events within most tumors, particularly BRCA2 is an early event. So what we've come to learn is that for BRCA2 and probably CDK12, you could sample any piece of the tumor and you'll get probably not the identical profile because there'll be subclonal changes, but the same deranged class of genome. For some other alterations, it is much more variable. So RB1 defects, that's not a DNA repair gene, but it's a common class of alteration that can occur actually after metastatic colonization. So then you may start to see differences between primary and metastases. And of course, the androgen receptor is only altered after we apply treatment. So you tend to only see those alterations in the metastatic setting. So it's a little bit context dependent, depends what you're looking for. Just to indicate a few other examples, we've shown, published a few years ago, that TP53 alterations are strongly prognostic, independent of clinical factors in CRPC. AR amplification shows some evidence of a differential relationship when patients are treated with chemotherapy versus ARPI. Not everything is bad. SPOP mutations, which are present in about 10% of patients, seem to have an association with good outcomes. And then here's a case report showing that rare alterations can also still be relevant. This is a patient with an AKT1 hotspot mutation showing who derived benefit from monotherapy of patasertib, which is an AKT inhibitor, despite having heavily treated CRPC. So, all these different alterations kind of, I hope I'm just kind of building up a picture that suggests like, okay, we have this emerging evidence that these different types of genomic alterations in prostate cancer are worth detecting. One kind of technical thing I'll highlight is that in order to find those alterations, you have to have ctDNA present. So that is a key limitation of the liquid biopsy because not all patients have ctDNA. And then not all patients have really high ctDNA either. So many assays are kind of operating around slightly opaque limits of detection and often not necessarily reporting when a sample is actually not evaluable because the CT fraction is low. So we always ask ourselves, if we get a negative result, was there enough ctDNA present in the sample to find that result? If not, it's actually inconclusive, not negative. And that's something that the ctDNA commercial world is kind of slowly getting on board with, but they need more encouragement. A couple of other things to highlight about tests in general. This actually came up on our molecular tumor board last night for the GUNS trial. And I was just having a conversation with Martin Gleave and co about, essentially, a bit of a failure in the way many commercial tests currently report is that you'll often get these long lists of mutations, sometimes in genes you've never heard of. We're often on the molecular tumor board kind of Googling genes on the side, what is this? But the reality is that most of these tests are reporting mutations, but not deletions, not copy number changes. So if you imagine you've got a panel of about 500 genes and they've reported 5 mutations, well, how many of those other genes actually have lost one copy through deletion? In prostate cancer, which predominantly is copy number driven, it's likely to be considerably more. So we have this sort of weird situation where we're focusing on these few mutations, but unaware of all these copy number changes that are occurring. And I would highlight that, in research, in academic research, copy number changes have been routinely reported for 15 years in tissue tests, right? The original TCGA efforts all report monoallelic, biallelic deletions, gains, and so forth. So it isn't necessarily a technical challenge, it's a policy, I would say, at the moment. The other kind of interesting caveat to think about, and I just mentioned this because just to highlight that everything we do is operating in a gray zone, right? There aren't these obvious binary always these binary decisions we can make with regards to biomarker status. You often hear about, let's say, HER2 amplification, AR amplification, EGFR amplification. I just want to remind that this is actually a continuous spectrum from low to high, right? We're looking here at some of our data showing the different copy number levels of HER2 in patients with bladder cancer. And so some of these have the equivalent of a 5 copy gain, others a 10 copy gain, others a 50 copy gain. And I just kind of often ask the questions of people, should tests just report HER2 amplification in this sample, or should we actually be differentiating between a 50 copy gain versus a 5 copy gain? Because I'm going to bet there's a biological difference there. The last technical thing I'll highlight is clonal hematopoiesis of indeterminate potential. You may remember when I showed that one of the slides at the start of the different things that can contribute to cell-free DNA. One of the key contributors is expansions in the hematopoietic population, especially in elderly cancer patients. And in fact, the deeper you sequence white blood cells, basically you'd find that everybody above the age of 70 will have some expansions in their hematopoietic populations. So those populations are contributing to cell-free DNA. So mutations in the white blood cells are present within your cell-free DNA. And we know that sometimes these mutations can fall in genes like ATM or TP53 or even BRCA2. So it's really important that you be able to recognize the difference between a mutation that's coming from the CHIP populations and those that's coming from actual ctDNA. It's a really easy way to do that, and it's profile match white blood cells in all patients. Not all tests are doing that. You need to be aware of those that are not. So what we do in prostate cancer and bladder cancer, first of all, we always sequence match cfDNA and leukocyte DNA from the same sample. We pull down a shallow whole genome, essentially, so we get that genome-wide kind of picture. We profile introns and flanking regions around genes so we can get the precise copy number level. We also can look for chromosomal rearrangements. And this panel has been being applied in a range of different studies. One I'll highlight is a prospective phase 2 trial we've been running across Canada, where 11 sites are sending blood for ctDNA screening here in Vancouver. And on the basis of the genomic profiling report, this is metastatic castration-resistant prostate cancer, these patients are then stratified onto different treatment arms. And we've actually now screened, I think, more than 500 patients across Canada over the last few years. And these patients get matched with different treatments depending on the molecular profile of their ctDNA. And actually, Ed Kwan in the audience, who's also a medical oncologist as well as a PhD and postdoc now in my lab, he's been summarizing the genomic landscape of these patients. We're hoping to put something out soon on the first kind of preliminary results from this trial. So the final thing I'll talk about in the last couple of minutes is a little bit about defining new biology and resistance mechanisms. So here we're not necessarily directly about to impact the clinic, whereas I think some of those other things I've mentioned are kind of much more ready for prime time. Now, how can we use ctDNA to understand what's going on in our patients that are progressing? One result I'll highlight, we reported now 5 years ago, the first example of genomic resistance to a PARP inhibitor in prostate cancer. And so this was a patient with a germline BRCA2 mutation that disrupts BRCA2. And this patient had a clinical response to PARP inhibitors, to a PARP inhibitor, but he then developed progressive disease after 6 months. And when we looked in the liquid biopsy, we could see a range of different reversion mutations. So a reversion mutation is when the cancer itself has corrected or literally reverted the mutation to restore BRCA2 function. So it's actually, normally it's a small deletion that cuts out the original mutation and restores the reading frame of BRCA2. And so in a patient with a germline mutation, what that means is the cancer becomes the only cells in the body that don't carry the germline mutation. That's wild that this can happen in just 6 months. And not just happen once. In this particular patient, in the liquid biopsy, we saw 7 different reversion mutations, so suggesting that different metastatic sites figuring out different ways to do the same thing. So this summer we showed that when we see different types of alterations, it's generally reflecting actually different tumor populations that are present in ctDNA, probably from these different metastases. And when you have a whole genome, you can actually deconvolute the different populations that are present here. And without going into too much detail, what we see is that if we sample patients over time, we see changes in the prevalence of these populations. So you can kind of imagine in this situation on the right-hand side here, middle right, we had kind of 3 populations at baseline. By time point 2, we can see pretty much the same thing, but time point 3, this rapid expansion of a new population emerging, and this would probably contain, in one particular case, a series of BRCA reversions or so forth. But ultimately, what we see is that even in the late stage setting, even after multiple lines of treatment, there's still concerted evolution going on between within the patient. So the genome is continuing to shift over time. In metastatic prostate cancer, the shift tends to drive increasing changes of the AR gene locus. So even if you already have an AR amplification, what you'll have after you apply more selective pressure is an even higher AR amplification. So you'll continue to drive evolution at the loci that you're targeting. We've seen that many times. This particular patient went from 20 copies of the AR to 50 copies of the AR after 2 lines of AR pathway inhibition. So the final thing I'll mention within this section and then wrap up for questions is about going beyond the DNA sequence itself. So you may have heard that it's possible to do methylation profiling of cell-free DNA or even ChIP-seq directly from plasma. Now, things like that do seem to be providing some epigenetic pictures about the cancer, but it means you have to do another library prep technique. You've got to go back to your sample, do another round of processing and so forth. So we asked the question, can we get some of the phenotypic information from the DNA sequence we already have? And the reason this is actually theoretically possible is that ctDNA and cell-free DNA has gone through apoptosis. So when we get a tissue sample, one of the first things we have to do with the DNA is chop it up into little pieces so we can actually sequence it. It doesn't come pre-fragmented for us. But the blood DNA has been processed by the cancer cell itself. And the apoptotic machinery cuts according to the chromatin landscape, the epigenetic landscape of the DNA in that cell, right? It preferentially chops in between nucleosomes. So what this means basically is that you can infer from ctDNA sequencing where the nucleosomes were in your cancer cell. So why would this matter? Well, what we did was we generated per sample nucleosome occupancy maps, and then we correlated those with whole transcriptome sequencing of same-day biopsies from those patients. So we had the ctDNA nucleosome maps, and then we had RNA-seq from a metastatic biopsy of the same patients. And we saw amazingly that any highly expressed genes displayed features in cfDNA that were consistent with active transcription. So the best way to think about this is that if you have an actively transcribed gene, you think about the transcriptional start site of that gene. The nucleosomes have to get out of the way so that the transcriptional machinery can bind and initiate transcription. Whereas if that gene's not expressed, the nucleosomes can be kind of randomly distributed there. So straight away, that means that if you have a really highly expressed gene, you can actually see the difference, right? Because you're seeing no nucleosomes, no evidence of nucleosomes around that start site. And so that's kind of what you can, in the top plot, which is highly expressed genes, that sort of heat map is showing nucleosome order, and you can see these nice grooves, whereas down at the bottom, those grooves have kind of disappeared. So why would this, what could we use this for? Well, the other place in the genome that nucleosomes would get out of the way is when the androgen receptor needs to bind. So the androgen receptor is obviously a key factor within prostate cancer. It binds across thousands of different regions in the genome and then initiates transcription of those genes. In this particular experiment, we looked at over 3,000 binding sites across 80 odd ctDNA samples from prostate cancer, some neuroendocrine prostate cancer, some bladder cancer. And basically what we saw is that most samples suggest that we have this nucleosome depletion signal at AR binding sites. So that's consistent with what we'd expect in late stage prostate cancer, continued evidence of AR activity. But there was this curious set of samples that had much lower levels. And they tended to be those without AR gene augmentation. They tended to be in the neuroendocrine prostate cancer samples, and of course also the bladder cancer samples. So what this is telling us is that potentially we can do, at the same time, find out the androgen receptor genotype. Is it amplified? Is it mutated? Is it rearranged? And also get a bit of a readout of whether the AR signaling pathway is active in that individual from the same bit of data. My final slide is a kind of case example of how this can be helpful. This was a CRPC treated with enzalutamide followed by abiraterone. This was actually in a clinical trial before we knew that sequence of drugs is not particularly effective. What we saw is over time, this yellow population, which was the dominant population in ctDNA to start with, we saw that kind of disappear over time, and we saw a new population represented in blue emerge. Now, this blue population had an RB1 mutation. And some of you may know that RB1 loss in conjunction with TP53 loss is associated with lineage plasticity. And so then we looked at the nucleosome depletion signals around AR binding sites. And we found for the baseline sample, this yellow population, we saw, sorry, the pointer stopped working. The deep red curve on the right-hand side is representing massive nucleosome depletion at AR binding sites, indicating that we had a really strong AR activity within this sample at baseline. But at second progression, the blue line indicated by the black arrow, we see almost no evidence of AR binding across the genome. And this is just over the course of 5 months here. So what we were probably seeing was the emergence of a AR negative population, and this patient was subsequently biopsied and found to have neuroendocrine prostate cancer. So, I think really a case report that illustrates the potential for ctDNA profiling to give us genotype and phenotype in the same test. And our ongoing discovery is based on a very large biobank where we have now more than 1,500 patients that are providing regular samples, and so we're hoping to kind of refine our observations through this biobank. And thank you to everybody. I think I have 10 minutes for questions.